Okay, we are now recording. Go ahead.
All right. Hello, good morning, everyone. Thank you for joining us on this kind of anomalous water issues seminar, which is also kind of cross-pollinated with our PFAS lateral team meeting for the month of January 2026. We've got Dr. Heather Whitehead with us today, and she is going to talk to us about some non-targeted analysis work that involved PFAS in surface and groundwaters. So Heather Whitehead, Ph.D., is a research scientist at UL Research Institute's Chemical Insights. And she used targeted and non-targeted analysis workflows to support the investigation of legacy and emerging contaminants. Prior to joining Chemical Insights, Heather was a physical scientist at EPA's ORD, which I think most people are familiar with, where she developed publicly available tools and workflows for non-targeted analysis, including a Tails toolkit for PFAS. Her doctoral research at Notre Dame focused on developing highly selective analytical methods for compounds relevant to human health and the environment, including innovative applications of particle-induced gamma ray emission spectroscopy for rapid PFAS screening. Wow, I'm really glad that I read that correctly the first time through. So thank you so much for being with us, Heather. Super appreciate your time, and please feel free to take the floor.
Perfect. Well, thank you for the introduction, and thanks everyone for joining. I saw some friendly names, I guess, of folks that I've worked with, excuse me, at MPCA and Department of Health. So it's really good to be back with you all in some capacity. So as the introduction alluded, my background is really in targeted and non-targeted analysis of PFAS, among other environmental contaminants. And so that's what I'm here to talk with you about today, is the specific study that we did in collaboration with MPCA and Department of Health to utilize non-targeted analysis to really support this investigation of surface water and groundwaters with known PFAS impacts. And so I'm currently employed with UL Research Institutes in their Chemical Insights division. But because at one point in time when this work was done, I was an employee of the Environmental Protection Agency, got to include the classic EPA disclaimer that anything that I present today are my own views and not necessarily those of the US EPA. But so really what I'd like to work through in today's talk is kind of three separate areas. And the benefit that I have is that all of the work that we did with Minnesota has been published. And so we're relying on these three key publications to kind of walk through our results. And so I'll start today with more of this background on non-targeted analysis, what it is, what the workflows look like. And that's through a publication that described the Entails toolkit that we developed. From there, I'll move on into our specific application of non-targeted analysis here in Minnesota, looking at surface and groundwaters that were impacted by historic PFAS waste sites. And then we'll wrap up today with our final kind of tidbit of work, which is taking those PFAS that we identified in our first study and using our kind of novel quantitative non-targeted analysis workflows to estimate concentrations of novel PFAS present. And so I'll cover all three of these. If you'd like to know a little bit more, there's the links to the DOIs. And like I said, I'll include a PDF of the slide deck after, so you don't have to try and race and find them now. But yeah, so the most logical place to start is kind of this background on NTA workflows. So let's just jump into it. And so I think everyone on the call is already familiar with this concept, so I don't really want to belabor the point other than to say that kind of we all recognize that modern commerce has introduced a large number of chemicals into our environment. And because of the way in which manufacturing occurs, a lot of the things that are introduced into the environment are chemicals that we don't know a lot about. So there might be novel structures, there might be structures that we can define, but we don't know a lot about their physical properties, and we don't know a lot about their toxicity or hazard. And so one way we can kind of think of this is like an iceberg, where everything above the water, the tip of the iceberg, are things that are chemicals that we can describe nicely, right? We have these defined structures. We can say something about their toxicity. We can say something about their environmental presence. But certainly for PFAS and really for most chemicals, much of that chemical class is going to exist beneath the water. Things that we don't know their structure, we don't know their toxicity, and we cannot at all define their environmental presence. And so one way we can characterize this is with our traditional targeted analysis methods that we use for PFAS analysis. And so I think most folks will know that at most those are going to include 100 compounds, but on a routine basis, maybe up to 40 compounds. And that's a fraction of the PFAS that are expected to be both structure-wise predicted and known, and also those that are present in commerce, those that may actually enter the environment and that people may be exposed to. And so as an analytical chemist, I focus on kind of this pressing need for analytical methods that allow us to more comprehensively identify those PFAS, and not just identify them, but also quantitate them and say something about their structure and the impact that may have on exposure to them. And so as an analytical chemist, I just kind of want to orient you to the differences in our non-targeted analysis methods relative to the traditional targeted or suspect screening methods that people are more familiar with. And so if we start over here on the left, our traditional targeted analysis methods that are commonly used in public health laboratories are excellent for a variety of reasons. The downside of those is that they're going to focus on relatively few chemicals, and they're going to be chemicals that we're going to predefine at the start. So we're going to want to know, or we're going to have to know immediately what their structures are. We're going to have an analytical standard and reference data available for them. And because we have this kind of plethora of reference data and analytical standards available for these few chemicals, it means that the results are relatively harmonized. One laboratory can produce the same set of results the next laboratory can. That makes the results really transparent. It makes it easy for one lab to interpret or review the results from a different laboratory. And that transparency and harmonization means that those results are relatively defensible, right? These types of methods are regularly used in regulatory and screening work. And so this means that you can pick up the results, apply them to a regulatory statute, and have confidence in those results that are generated. And so we move up the scale one more step, and we work towards these suspect screening workflows that are similar to targeted analysis in that they have relatively few chemicals, few being hundreds of or thousands of chemicals per library. And those chemicals are relatively predefined. Though importantly, these chemicals, we may not have an analytical standard for them in-house, but we're going to rely on this library data, a spectral library, a retention time library, to inform both the chemical structure and the measurement properties of that chemical. And so these are nice because while we may not have that data in-house for all, say, 1,000 of those chemicals, we have a plethora of reference data available that supports their measurement. And that means to some degree we can use suspect screening approaches in routine analysis workflows and also in more research and investigation workflows. So the best example of our suspect screening workflows that most folks are probably familiar with is a GCMS. A lot of GCMSs or gas chromatography mass spectrometers, they come with a NIST library of maybe 10,000 chemicals in them with spectral library data. And so that would be considered a suspect screening method. You don't have a standard for all 10,000 chemicals, but with some degree of confidence, you can say that that is that chemical. The methods I'm talking about today are non-targeted analysis methods. And the beauty of these methods is that we, in theory, can look for any chemical. We're not subject to a predefined list of chemicals. And we're also not going into it knowing what the structure of that chemical may be. And so we can look at a much wider chemical universe, and we don't have to say something about that chemical before we go and measure it. The downside of this then is that if I don't know what the chemical is, I can't guarantee that I have an analytical standard available for it. And so if we measure a novel chemical, the likelihood that there is a pre-existing analytical standard is relatively limited. And that means that the results are a little bit more opaque. I can't transfer a non-targeted analysis method to the next laboratory and you immediately be able to interpret or utilize those results if you don't already have some kind of existing background or expertise in non-targeted analysis. And so historically, our non-targeted analysis methods really are restricted to the research and investigation space and haven't routinely been used in public health laboratories or other places where more routine turnaround work is done. And so to kind of push us towards this idea that non-targeted analysis is a useful tool in these public health or routine laboratory spaces, I just want to show you what a typical NTA workflow looks like. And if you're someone that works in a lab or that does targeted analysis, you'll know that these are the exact same steps that you might do in a targeted analysis study. So you have to design the study, you have to then go out and get your samples, prepare them, and then analyze them on the instrument. And then once you have them acquired or their data acquired on the instrument, you can process, analyze, and report back that data. So all of that is true of a non-targeted analysis study. These three areas, though, the data acquisition, the data processing, and the reporting and communication, are really areas where a non-targeted analysis study begins to differ, and where there's kind of these subtleties that we have to consider before we engage in non-targeted analysis. And so I'll cover the kind of three areas here in some detail, though at the end I'll point you to our Entails toolkit, where if you want to see a full description of this workflow and how non-targeted analysis differs, that would be the best place. So let's start by kind of setting some boundaries for non-targeted analysis, since most folks are likely unfamiliar with the technique. So a term you will hear often for non-targeted analysis is this term chemical space. So we can define that as the constituents of a sample across a multidimensional swath of chemical properties. And so what I mean by that is that chemical space can almost be represented like a universe that you see over here on the right. And in the universe is every chemical that could be measured in that sample. If this were a perfect world, I could measure every single one of those chemicals present in that sample and not have to do anything to get that sample ready to be measured. In reality, the folks that work in the lab know that it takes a lot of time to take a sample from the environment and prep it for instrumental analysis. And so here's an example of all the different types of steps that may be included from taking the sample in its true natural form into the detectable space that we measure on the instrument. And so things like, how you extract the sample and what type of filter you use are all filters that act as a limitation on this chemical space. And so we move from this wide universe of all these chemicals that you might measure within that sample to this more narrow detectable space. And so each of these steps is itself a filter. And so when you say I'm using non-targeted analysis to measure all the chemicals in this sample, what you're really doing is saying I'm using non-targeted analysis to measure this particular detectable space based on the choices that I'm doing. So that's an important caveat for NTA. We're not measuring every single chemical in the sample. We're measuring what we kind of are allowed to based on the prep methods that we use. And for folks that work a lot in PFAS, you kind of already recognize this observation. So I'm using a figure here from a 2018 publication that showed the different types of analytical techniques that we might use for PFAS analysis. And so they work from very generalized methods. What I did in my graduate work, or a technique I used in my graduate work with this PIGY, particle-induced gamma ray emission spectroscopy that measures total fluorine, both inorganic and organic. And that contrasts with our targeted traditional PFAS analyses using liquid chromatography tandem mass spectrometry. And so I'll point out that our non-targeted analysis methods kind of exist here in this orange-ish color LC HRMS box. And so they expand our chemical space, but certainly for PFAS, they're not going to tell us everything about what's in a sample. They're not going to include all forms of extractable organic fluorine, and they're not going to include other forms of fluorine, particularly polymeric, that just aren't amenable to the techniques that we're utilizing. And so for PFAS analysis, our non-targeted analysis methods are powerful, but they're not going to tell you every single thing. So that's just another quick caveat. So our non-targeted analysis methods, as the last slide suggested, are going to utilize high-resolution mass spectrometry, or HRMS, to measure what's known as a mass-to-charge ratio of an ion. And so I'm showing here, excuse me, a simplified schematic of a high-resolution mass spectrometer. And so we've got kind of three main stages here. The first of those is our sample separation technique. And in all of the work I do, that's going to be liquid chromatography. So on these sample separation techniques, what you're doing is you're giving the instrument a complex mixture of chemicals, and you're asking it to separate out those individual components. And so for each individual chemical, you're going to get what's known as a retention time, which is the time that that chemical elutes. From there, once you have your chemical components separated using chromatography, you have to generate an ion. So as this suggests, a mass to charge ratio. The charge is the important part there. Our mass spectrometers need a charge to be able to measure a chemical. And so we're going to take that chemical and ionize it. That can either be a positive ion, like what's shown here in the image, or it can be a negative ion. Once we have an ion, that's what we actually are measuring. And so we send the ions to our mass spectrometer. And what we're asking our mass spectrometer to measure is this mass to charge ratio. And so, as the name would suggest, it's both the mass of the ion as well as the charge. So an ion can be multiply charged, a 2+ or a 2- charge, for example. The beauty of our high resolution mass spectrometry techniques is that when we measure these masses, like down here, you can see we get what's called an accurate mass. This mass is reported out to 4 decimal places. And that 4th decimal place going out that far is incredibly powerful. And the reason for that is, as you can see, we have two masses here that are actually relatively close to one another. But because of the use of these high resolution mass spectrometers, we're able to tell those two masses apart. And when we're talking about trying to identify a chemical and its chemical formula, those minor differences in mass actually have a big impact on the potential chemical that can be. And so ultimately what we're getting from these instruments are both that retention time measurement and the measurement of its mass to charge. And so what we actually are measuring in non-targeted analysis is that measurement of a mass at a specific retention time. And so we're broadly going to call that a chemical feature, or sometimes just a feature, and that is a mass at our retention time. And so what I want to point out here, though, is that a chemical feature, that representation of a mass at a retention time, is not necessarily a chemical in itself. So a given chemical can actually produce a lot of chemical features. And so on the right here, I have an example of a well-known PFAS, Gen X, and the parent structure is shown here. If you do a lot of PFAS targeted analysis work or utilize the data, you might actually already know this, but when we measure this guy using, for example, EPA method 1633, we aren't measuring the intact parent molecule shown here. What we typically measure is actually this first fragment ion that has the loss of this CO2 group. And the reason is because when I put this guy in the mass spectrometer, it prefers to make this fragment. Gen X, though, makes a variety of things when you put it into the instrument, not just this first fragment. It produces, at the time of ionization, a few different fragments. It produces the monomer just in much lower abundance. Then it also produces these other things like dimers and dimer adducts. And so this one chemical can produce a variety of different things that we can measure from it. And so each of these would be a representation of a feature, this mass at a given retention time, but importantly, it is not a unique chemical. It is a feature that is related to this one parent chemical. And so when I say down here that a single NTA study can have thousands of chemical features, that's not to say that each sample has thousands of chemicals present in it, just that those chemicals that are present in it can produce a variety of features. So in terms of NTA data acquisition, the biggest part of what we're trying to acquire, is our mass data. And so there's two primary mass measurements that we're trying to acquire with our non-targeted analysis methods. The first of those is to get what's known as a precursor ion M/Z value, and that's colloquially going to be called the MS1 level data, so MS1. And so what the MS1 data helps us do is restrict the possible chemical candidates, so restrict the possible chemical formulas that that ion could be. And so in this particular example here that I have on the right, we have the mass spectrometer and it's measuring three precursor ions at the same time. If we focus in on just precursor 1, let's say precursor 1 has a measured M/Z value of 412.9642. We can return a list of candidate formulas, and we can't say anything beyond that, right? We have these three different formulas, a C6H5F6NO13, for example, and its theoretical mass is very close to our measured mass. There's a very small mass difference there. We can also have things that are a little bit further away. So the C8F15O2, for example, just is a little bit further away. And then we can have things that are even further away in terms of their mass differences. And so this alone, we wouldn't be able to say anything more specific about which of these is the most likely candidate for this 412, but we can say, here's the list of potential formulas that it could be. For this scenario, I'm going to point you to that this mass is actually B right there, and it would be the structure of PFOA once ionized. So if it were the structure of PFOA once ionized, the way in which we would be able to actually definitively say that is by using the secondary mass measurement data, which is its fragment ion M/Z values, or the MS2 level data. So our MS2 level data is going to be able to tell us more about the substructure of the different of the chemical feature that's presented. And so this might tell us things like what functional groups are present, where certain bonds are located, and that ultimately helps us take those chemical formulas from the last slide and then propose a structure. And so in the example of precursor 1 over here, if we take it, we fragment it up, we might measure these three separate fragments. Those three separate fragments are shown here. And then for these fragments, like what we saw on the last slide, we can propose candidate formulas for those fragments. And so in this example, we have a fragment at 118 that might be tied to C2F5, and we can draw that out on this structure up here, and we keep going upwards. And so the goal of these mass measurement methods are to get both this detailed structural information about what fragments are present and how they relate to the overall structure, and then also the total structure to propose a correct chemical formula. In practice for our non-targeted analysis methods, we're doing a lot of this at the same time. And so at any given moment, the mass spectrometer is measuring a lot of MS1 level data. And then for a select number of features in the MS1 level data, we're also collecting that fragmentation information. And so in a given NTA acquisition, we can get thousands of chemical features measured through the MS1 and maybe a few hundred things where we measure the MS2 level data. And so it's the combination of both the MS1 and the MS2 level data that allows us to say something about the structure of a particular chemical feature. So I hope this impresses upon you that we generate a lot of data with non-targeted analysis, and it's not like targeted analysis where you can immediately tie it back to an individual chemical. And so a big part of non-targeted analysis is how you process that data to make meaningful interpretations out of it. And so, you know, for an NTA data processing, each instrument vendor, whoever you buy the instrument from, has a really unique set of software and algorithms that translate their raw non-targeted analysis data into meaningful chemical identifications. And the folks that work in the laboratory on these instruments know this. Some vendors are better than others, and we could talk about it all day long. But for the work that I'm describing here, what we really wanted to focus on was tools that were vendor neutral and open source. And particularly in Minnesota, you actually have a handful of different non-targeted analysis instrumentation. And so for this work, we wanted to be able to utilize one tool that could take in all of that data from different sources and process it using the same set of parameters and modules. And so the workflow that I have over on the right, I won't go into detail on. If you're interested in it, I can talk more offline sometime, or I can point you to the Entails toolkit. But essentially, we use this workflow that allows us to aggregate information from publicly available databases and resources, things like EPA's CompTox, other mass spectral library databases that both EPA and other resources are developing, and allow our identifications to be supported by kind of robust chemical information that's available in the public domain. So once we worked from our raw data to identified PFAS, a big part of NTA is how you then choose to report that data. So if it hasn't kind of been captured yet, there's a lot more uncertainty associated with the chemicals that we identify in non-targeted analysis because we don't have things like those analytical standards or often a plethora of that reference data available. And so anything that we identify with non-targeted analysis methods has some inherent level of uncertainty associated with it. And so it's important that we communicate what that uncertainty is ahead of time. And so the system that we commonly use for how we report that uncertainty is shown on the right here. It's commonly referred to as the Szymanski scale based on the authors who developed it. And so the way the scale works is working from level 5, our lowest level of confidence, all the way to level 1, our highest level of confidence. We build in these different tiers of supporting data that we acquire as part of our non-targeted analysis study. And that allows us to work from just simply the mass that we measured all the way up to a confirmed structure where we have a reference standard available and we can say definitively this is that chemical. For a lot of non-targeted analysis studies, we work at these level 2 and level 3 identifications, places where we may have some reference data available and we can say something about the structure, or places where we may have no reference data and we can propose these tentative candidates where we think we know the structure, but there's maybe some uncertainties that we can build in and define more clearly. And so I'll show how we use this in practice when I show you the actual results from Minnesota, but just to orient you to the existence of this schema. So as I mentioned at the start, there's a lot more here that I have not covered about the differences for non-targeted analysis. What are the best practices for how we do non-targeted analysis specifically for PFAS? And so for folks that are interested in learning more about non-targeted analysis or how it's applied to PFAS, I'd encourage you to go look at our toolkit where we kind of captured all of this. It is a ton of content and we worked really hard on it. That link should take you to the website, it should actually direct you to the newest version of it. Like I said, I'm no longer with EPA, and so they've taken it off my GitHub and put it on a different one so somebody else can maintain it. But it should still take you there. And yeah, this covers everything, and we show everything that we did as part of these studies that I'm going to talk about in more detail. So switching gears now and focusing on the specific work that we did here in Minnesota for utilizing non-targeted analysis in impacted surface and groundwaters. And this is the best audience to give this presentation to, right? Because I don't have to describe to you all the complex background and information about PFAS contamination in Minnesota. You guys are way more educated on this topic than even me. So I'm going to fly by some of the background here and just point out that obviously PFAS contamination, specifically within the Twin Cities, is to a large degree driven by the industrial research, manufacturing, and waste disposal from 3M. For this particular study, we're focusing on 2 sites and their surrounding areas, and that's the Oakdale disposal site and the Washington County landfill that I'm certain all of you are very familiar with. And so the scope of this work was that MPCA, Department of Health, and the other agencies obviously had a plethora of information available about PFAS contamination as it relates to targeted analysis. But there was the big question still of what other PFAS may be present in these sites and their surrounding surface and groundwaters. And so that was the scope of our study. And so the non-targeted analysis study that was developed for this work was again focused on those 2 waste sites, Oakdale disposal site and Washington County landfill, and surface and groundwaters upstream of those sites, at those sites, and downstream to those sites. And so there was kind of 3 separate goals. The first of those is to identify additional PFAS that may be present that are associated with these particular disposal sites. So again, that's collecting samples that are upstream, at, and downstream to those sites. Once we identified the PFAS that were present, we wanted to be able to utilize our quantitative methods to actually estimate their concentrations that were present in the environment. And then lastly, we wanted to be able to say, if a PFAS is identified at these source sites, is it present at any of these downstream sites? And what is the fate and transport of those chemical contaminants as they move throughout this particular watershed? And so I'll plug it again at the end, but if you want to know more about these particular sites, I have, as part of our publication, a big GIS file that shows their exact latitude and longitude. I know a big question will be, for the groundwater, what were the wells? And so all of that is packaged in a GIS file. And I'm certain the folks at MPCA who we worked with can also pass over that information to you, should you be interested. So I'm going to breeze through kind of the data generation and processing here because it's not the fun, juicy bits of what we want to talk about. But I will point out that this collaborative study was done between EPA's Office of Research and Development and the Minnesota Department of Health and Pollution Control Agency. And so I had the lovely benefit of basically telling Department of Health and Pollution Control Agency what the best ways for acquiring their data was. And they were the ones that actually acquired all this data. So samples were collected, prepared, and acquired by Department of Health. They used EPA method 1633 as kind of the baseline. And in fact, a set of samples were also acquired for paired targeted analysis on these same sites. So your friends in Department of Health generated all of this data, and they're very skilled and excellent people to work with. All of the samples that were analyzed were injected in triplicate alongside an extensive amount of QA/QC. And we used our non-targeted analysis methods in negative ionization mode only. That doesn't mean a lot to you unless you're a really intense analytical person. But what I'm trying to point out there is that we only measured PFAS that are going to be kind of anionic and, in the typical sphere of what we think about as PFAS. So PFAS that maybe are positive or zwitterionic are not going to be captured in this particular method. And so the image on the right here is kind of a repurposing of the data processing workflow, and it shows each of the steps that the data underwent. So we had those 17 study samples. There's actually one more sample of the SAF concentrate from Tablin Park, but it wasn't a huge focus in our study, so I actually won't talk about it at all today. So 17 plus 1 samples. From that, we got about 7,000 chemical features. And from there, what we were able to do was process the data through a variety of MS1 and MS2 level data annotations. We do a lot of QA/QC filtering and PFAS-specific annotation workflows. And what we landed with were 250 chemical features that had an annotation, and 210 of those were identified to a unique PFAS. So 210 unique PFAS that were identified in these surface and groundwaters. And so as I promised, this Szymanski scale schema would return. And so here's those 210 unique PFAS broken down by the confidence levels at which we identified them in. And so just as a reminder, our level 1 confidence level is our highest level of confidence. So things that we can confirm with our reference standard. And as we move down all the way down to 3D, these are things that are less confident, though we can at least propose tentative structures for these particular compounds. And so things that we identified at level 1 were those PFAS that were already included in EPA method 1633. So the targeted method that was employed alongside the sample set. 32% of the compounds that we identified are at this level 2 confidence level. And so these are going to be chemicals that we can say definitively this is the structure. And we have some available reference data to support that assignment. And then the lion's share, 52% of our compounds exist in this tentative structure space. You'll see on the subsequent slides, we have a lot of identifications where we feel pretty good about the chemical structure, but maybe the difference in a bond location is what it's impacting our ability to say definitively this is the structure. And so you're seeing on this slide, I'm often referring to this idea of a homologous series as part of our defining characteristics. So it's probably worthwhile that I define what a homologous series is. And so if you do a lot of work in PFAS, you've probably heard this term homologous series before. A homologous series is this idea that for something like PFAS, where you have a base structure like PFOA, for example, it's got a carbon-fluorine backbone. And from that carbon-fluorine backbone, we can remove or add other CF2 subunits. So if we add a CF2 subunit, we go up to PFNA. If we remove a CF2 subunit, we go to PFHPA. And so in the measurement world, we can rely on these homologous series to help us actually identify PFAS because they make a nice little relationship when you measure them. So on the instrument, their mass will increase by that same CF2 unit, and then their retention time will also be characteristic. They will tend to elute within a set period of time after one another because the addition of the CF2 means that it elutes a little bit later. And so we use homologous series as part of our main workflow to help us identify PFAS in these samples. And when we look at these particular homologous series, we find 33 unique homologous series that represent 196 individual PFAS. And so a lot of homologous series were identified in this, and I'll show you on the next slide what those structures look like. But I want to point out that for almost all of these homologous series, the chain links of that fluorinated backbone tended to range from C3 to C10. So if you're up on your PFAS history, you know that C8 is kind of the historic PFAS, PFOA, what we think of. In today's day and age, we've shifted to shorter chain chemistry, so C3 to C6, let's say. And historically, longer chains of PFAS all the way up to C10 were commonplace too. And so what were the structures of those homologous series that we've identified? A big bulk of the homologous series that we identified were the sulfonic acid species where there is an SO3H head group on them. The individual structures within varies, and I'm not an organic chemist, and I don't want you guys to have to become organic chemists today, so I won't point out all of these individual structures other than to say that the bulk of what we saw was the sulfonic acid species. We also saw a handful of carboxylic acid species where now that head group is a CO2. And then we saw these other types, the sulfonamides and sulfonamido species, where we have this sulfur nitrogen containing group. What I'll point out about all of the species that we identified, they're all traced back to a specific sulfonyl-derived PFAS production process that 3M was the historic generator of. So 3M used this process pretty routinely, and I'll show you what that process looked like on the next slide. What I will also point out is that several of these homologous series that we identified, particularly the ones in the red box here, were chemicals that had not previously been identified either in the environment, and also their structures were not well described or existing in any databases. And so once we identified these chemicals, we had to then go and register them in the literature, and now they have CAS numbers and all the fun stuff like that. But they were not described prior to our identification of them. So as I mentioned, the bulk of what we identified were per and polyfluorinated sulfonic acid species. And so the process by which these species were made is something called electrochemical fluorination. And that is a 3M process through and through, was developed and utilized by 3M as the major ECF-derived sulfonyl derivatives for a variety of applications, things like AFFF, various coatings, semiconductor manufacturing, and much, much, much more. That process follows what's shown here on the right. So you start with a non-fluorinated precursor, the C8H17SH, for example. So a cute little thiol group on this hydrocarbon, but a pretty plain molecule otherwise. And then you subject that molecule to lots of fluorination so that you can produce this, sulfonyl fluoride group here. And then ultimately that takes you through another step to the final product. What is shown here is PFAS, but importantly, they can do this at any chain length they want. Obviously, since 2002, they have no longer engaged with this particular process as shown here because they've phased out the C8 chemistry, but they routinely do this for shorter chemistries and in the past also did it for longer chain lengths too. So as I mentioned, this is this particular process and the chemicals that we identified were very clearly tied to AFFF production, as well as other types of chemistries or end uses that you might find for these chemistries. And so let's look a little bit more now at the distribution of the PFAS that we identified within the sites that we investigated. So I'm showing here this crazy Venn diagram that shows the interaction of surface and groundwater at our 2 point sources. And so point 1 over here is going to be the Oakdale disposal site, and point 2 over here is going to be that Washington County landfill. So of the 210 PFAS that we identified, 208 were measured in surface and groundwater at either one or both of these sites. So the 2 that are missing there are actually 2 specific PFAS. If you're up on your targeted methods, that's in ethyl and in methyl fossa that we identified, not at the 2 point sources. Interestingly enough, those 2 chemicals are actually rapidly converted in the environment into other species, and they were measured at 1 downstream site. So that actually suggests another source that's leading to their input into the environment that's independent of these 2 point sources. If we look at these 2 point sources, though, we can walk through kind of the different distributions. So there's only 6 PFAS that were measured in the surface and groundwater at both sites. So these are going to be your legacy PFAS, things like PFAS, PFOA, and their shorter chain links on either side. There was 22 things that were measured in the groundwater at both sites, so at both point source 1 and point source 2. And these are again going to be traditionally historic type PFAS, things that are already measured through targeted analysis methods. We have another about 18%, so 19 and 18 chemicals that were measured in both surface and groundwaters at one or both of the sites. So definitely some relationship between the surface and the groundwaters. And what I will point out, the lion's share of the chemicals identified were actually unique to the groundwater at the Oakdale disposal site. So we measured 142 PFAS that were only found at that one site. If you're doing your math, you may be thinking, Heather, that's not very many chemicals that were measured at the downstream sites if there was 142 unique to just the groundwater at that site. And that is true. So 31 PFAS were measured at any of the downstream sites. So of the 210 we measured, only 31 were measured at a downstream site. And so what I'm showing you here on the right is called an upset plot. And in these plots, we're looking at the interaction of 2 things or multiple things to one another. And so on the bottom here, I've got the different downstream sites that we measured, sites that were associated with just one or the other point source. So point 1 groundwater, point 1 surface water, and then point 2 groundwater. And then these 2 in the middle that are our downstream sites where there's expected to be a mixture of both point 1 and point 2 impacting both the surface and the groundwaters there. And what we can see from this upset plot is that sites that are impacted by point 1, so things that are mixed or this point 1s, typically have much more chemicals present in them. Typically much more PFAS were identified in sites impacted by point 1, and this makes sense given that the bulk of what we identified was in our point 1 groundwater. The interactions decrease as you go down here, so there was very few things that were measured at all downstream sites. So looking here, just 1 chemical measured at all of these sites, and the bulk of everything was really measured in these sites related to point 1 and point 1 alone. So what PFAS were actually measured downstream? So we had a variety of chemistries. I'm not showing them all here. And again, I'm not an organic chemist and I don't want you to have to become one. What I'll point out is that a lot of these were the sulfonic acid groups. So you can see here that SO3 head group. And what I've done here is underneath each group, I've listed the fluorinated carbon chain link that was identified. And what you can see here is that a lot of these are that kind of middling C8 area. Since these are historic waste sites, things that were impacted by waste all the way, I think, from the 40s through the 70s, this makes sense. C8 was the largest produced PFAS at that time. And so the fact that we're measuring C8 specifically downstream is to some degree expected. And so I want to shift gears now and move on to the estimating of PFAS concentrations for these particular PFAS that we measured, so you can get a feel for kind of the magnitude or the scale of PFAS that we identified. And so first I'll start by kind of describing how our quantitative approaches for non-targeted analysis work, because they are a little different than what you might be used to if you're familiar with traditional targeted analysis techniques. And so, as I mentioned, we identified 32 PFAS that were part of that traditional targeted analysis method, so EPA method 1633 in this case. And so if we were using targeted analysis, we could do what's known as absolute quantification. And in absolute quantification, we can essentially take that chemical standard that we have for that chemical and generate a calibration curve where we've measured where we know rather the concentration of that chemical present, and we can measure the intensity. We can model that, and then we can say something about the relationship between the concentration and the signal that we measure. If we can generate a calibration curve, it means that we can measure an unknown sample. We can measure the intensity, importantly, of that unknown sample, and then relate that intensity back to a concentration and say for any given unknown at this intensity, it is going to be this concentration. And that's absolute quantification. For our non-targeted analysis methods, we don't always have analytical standards available for the things that we identify. If we don't have an analytical standard, we can't make this calibration curve for that chemical, and then we can't report back the concentration of an unknown. And so for things that we measure through non-targeted analysis, we can use what's known as quantitative non-targeted analysis approaches to ultimately estimate the concentration. So our QNTA approaches look something like this. What we're going to do is take all the chemical standards that we have available, in this case for our PFAS. So we took all 40 of those PFAS that were measured as part of EPA 1633, and we generated calibration curves for them that are, again, that representation of a concentration at its intensity or response. And so we're going to acquire that concentration response data. In practice, we're measuring the slope of that calibration curve for all of those chemicals. We're going to take the slope of all of those calibration curves and create this distribution that shows how all of these different PFAS, what their mean or median concentration response factor is. So we're going to make this distribution and we're going to say, hey, what's the 50th percentile or the median of that distribution? We're then going to take that median and say, let's apply the median response factor between that relationship between concentration and response, and say that's in general how PFAS are going to respond in this sample. And so now we have this median or this approximated concentration response factor, and we can apply that median response factor to our unknowns. So here you can see we have 3 unknowns, and we can then report them back to a concentration using this median approach. And so it's a lot more complicated than that. If you are interested in quantitative non-targeted analysis, I'd really encourage you to go read our paper that came out about a month ago now. It gets heavily into the math of it and how we actually ensure that we're doing this appropriately. I'm vastly oversimplifying it for the purpose of today's talk. So the results that we get when we apply these concentration estimates using QNTA approaches are shown on the right here. What I'll first point out is the range of concentrations that we observed over here on our y-axis. So this is 5 orders of magnitude that we observed concentration ranges in. This takes us from really low parts per trillion levels of PFAS present up to parts per million levels of PFAS. In particular, the groundwater at our Oakdale disposal site had some of PFAS concentrations at roughly 4 parts per million. I think you all are going to be well acquainted or adjusted to that idea if you've seen targeted results from these sites before, but it is still kind of a significant result in itself. So what we have here on this chart is a set of 4 different results. The black dots are actually the paired targeted analyses that were done on these samples separate of non-targeted analysis. And then everything in either red, blue, or purple are results generated from our QNTA approaches. The red shows the concentrations that we predicted for those same 1633 analytes. And so we can look at the comparison between the true targeted measurement in black with red. We importantly have the error bars or the uncertainty estimates that surround our QNTA estimate. And what you see is that all of our QNTA estimated concentrations fall really closely in line with what was actually measured through targeted analysis. So that's a key step of the work that we do. We always validate it back to some paired analysis. What we can also see here is that we've separated out the chemicals that we identified in non-targeted analysis that are at those lower confidence levels, so those Szymanski level 2 or 3s, versus all of the PFAS and versus those that are measured in our traditional targeted analysis with 1633. And what I want to point out to you is that these NTA Szymanski 2 to 3 levels tended to be significantly lower concentrations than their 1633 counterparts. And so the takeaway here is that the chemicals that we identified with NTA, so the PFAS measured only through NTA, represent about 10% of the total PFAS concentrations that were measured in these samples. So not a huge amount, but also not an insignificant amount when we're talking about total concentrations up to the parts per million range. So what does this look like for the individual sites here? The boxes are somewhat hard to see, but I've tried to represent it based on the different point sources. So everything in this darker blue here are things associated with the Oakdale disposal site. In this medium tone blue, things associated with the Washington County landfill. And then in this much lighter blue, things that are our downstream mixture sites where they're expected to be impacted by both. What we find is that our total PFAS concentrations that were measured were highest again at that Oakdale disposal site groundwater. And what we find is that as we move downstream, both within the Oakdale disposal system and within these mixture sites, concentrations tend to decrease at each of these sites. And so we go from significantly high parts per million level concentrations down to low parts per trillion concentrations at some of these downstream sites. When we look at the proportion of 1633 versus non-targeted analysis analytes, we see that most of these are dominated by 1633 analytes, but again, some of these have upwards of 10-20% of their total concentrations represented by non-targeted analysis only chemicals. So then the last question we wanted to answer now that we had this concentration estimation information is, what can we say about these PFAS that were measured, specifically those that were or weren't measured downstream, and focusing on the ones that weren't measured downstream? So for all of the PFAS that were not measured at any of our downstream sites, we really considered 3 scenarios as to why they may not be measured. The first of those was saying, well, does the particular PFAS that we measured not actually physically transport downstream based on its physicochemical properties? And our interpretation of that when we looked at the data was that most of the PFAS that we measured here are going to be water soluble. We measured water. If we can measure them, they're water soluble. And particularly the PFAS that we looked at are not expected, and from what data we have available, don't totally partition to other environmental containers. So thinking of things like soil and sediment, some of these PFAS would prefer to be in soil and sediment, but they won't 100% partition to those containers. And so there should be some movement downstream if it's there. So the second thing we considered was, well, maybe they're moving downstream, but as they move downstream, they're subjected to transformation or degradation, and we're not measuring that same PFAS anymore, we're measuring something else. And so what we found was that when we put all of the PFAS that we identified into the chemical transformation simulator that EPA's developed, what we find is that few of these PFAS undergo transformation. That fits within our sphere of understanding why PFAS are an environmental problem to begin with, right? They don't readily undergo transformation or degradation. There are a few PFAS that we detected that do undergo transformation, but importantly, they just transform to the traditional legacy type PFAS, things like PFAS, PFOA, and its shorter chain analogs. And so even if they are transforming, we're still capturing their measurement through other PFAS that we've reported in this study. And so then the third question was, well, maybe they're moving downstream, maybe they're not transforming, but they're actually being diluted as they transport throughout the system, and they fall below the limits that we can actually measure them using our instrumentation. And so what we found here is that when we model the dilution factors in surface and groundwater, there is significant dilution of these across the watershed. And so many of these chemicals do fall below the limit of detection. And so just to kind of show you what that looks like, what I'm showing you here is the measured concentration of a few different types of PFAS grouped here on the right by their kind of subclass. So we have the perfluorinated sulfonic acids, the perfluorinated carboxylic acids. These are sulfonyls, and these are keto-containing sulfonic acids. And so what we can see is that we have the different chain links for for the perfluorinated sulfonic acids, for example, we go from C2 to C10. We again see that our C8s are the largest, measured in the greatest concentration. And so I blocked out C8 in each of these. And so this is what we're looking at in terms of concentrations measured at the Oakdale disposal site in the groundwater. If we move then to just the groundwater at the next downstream site from Oakdale disposal site, we see a significant drop off in the concentrations present. So if I flip back between the two here, you can see the relationship of the chain links and the concentrations they were measured at holds true, but many of them are no longer detected at that first downstream site. And so what we found was that the estimated dilution factor from Oakdale disposal site to its next downstream site was about 650-fold. And so almost all of the PFAS that were measured at that groundwater at Oakdale disposal site are going to fall below our detection limits once they move downstream. And so it's not that the PFAS aren't there, it's just that they're below our detection limits that we can identify using these types of methods. So again, I'm kind of covering the highest level of all three of these works. There's a ton of work that was done that looks more into detail about, the PFAS that were identified, their use cases, what we can say about the manufacturing process, and again, a ton more about the math and the statistics behind how we do these PFAS concentration estimations. So if any of it interests you, feel free to ask questions or just check in with these publications. They're really stellar bodies of work, and I was really happy that we were able to publish them last year, given everything that was going on with federal research. What I'll point out to you is that if you go to these resources, we have a variety of data that's reported and available for you. What I'll say, though, is that all of the data that was generated is the ownership of Minnesota Pollution Control Agency and Department of Health. So Andrea Dalmier, for example, can get you just about anything that I could present on today. And so if you have any questions, feel free to reach out to me or her. We can get you the data. But within these particular publications, you can go out and you can find the full table that describes all of the PFAS that we identified. So all 210 PFAS, you can find out their formula, their structures. We have the unique identifier DTXSID to help you quickly link it to something like EPA's DSS Tox or CompTox database. We have the same exact thing for their concentration estimates in our Q&TA paper. So you'll find this exact same table, but appended to it will be all of the individual concentration estimates across each of the individual samples, should that interest you. If you are more on the analytical side, we've also supplied a detailed amount of the mass spectral data that comes alongside these identifications. So you can go in and you can look at the MS2 level data that we generated and see our annotation and interpretation of that MS2 level data. For all of the quantitative estimates, you can find those calibration curves. You can find the code that we use to generate the concentration estimates and a lot of detail about how we do that process. And then I know a lot of folks on the call are not analytical chemists at all, but are rather people that are working in the geology or hydrogeology space. And so we've included this ArcGIS file that allows you to look at the individual sites, find their associated metadata, and throw on a bunch of additional layers. So I have in there PFAS analytic tool data from EPA and a variety of hydrogeology and geology data that I found for the Washington County area. So NTA is great because you can identify new chemicals, but I think all of us recognize that just being able to say what chemical is there is not super informative rather, if you're trying to say something about its risk or hazard to exposure of that chemical. And so what I want to just spend my time wrapping up with is pointing out the downstream uses of NTA data. So how would we like to utilize NTA data to inform a decision? And so within the system, I think you recognize there's two unique sources and they have very different profiles. Source one in our example, Oakdale disposal site, was a very diverse source with lots of chemical signatures. whereas source 2 had fewer chemical signatures. And so in real world situations, we might want to use these different profiles of these sources to say something about how our downstream sites are impacted. And so one of the approaches that we're looking at utilizing at Chemical Insights, now that I've moved over, is taking our non-targeted analysis data and trying to map these source profiles so that we can say something about the contributions of these individual sources to our downstream sites and represent them as being, solely impacted by source number 1 or solely impacted by source 2 or somewhere in between. And so this would really help our exposure workflows and help us do source fingerprinting to inform more about our regulatory insights of non-targeted analysis. Then the other side of non-targeted analysis data is that I can identify a chemical, but if we don't have anything about its toxicity, it's difficult for us to say anything about hazard or risk. And so I think we all recognize that very few PFAS actually have traditional toxicology data available for them, PFAS being one of them. And that ultimately limits what we can do with these chemicals for hazard assessment purposes. And so our ongoing work at Chemical Insights seeks to take what we have in terms of traditional toxicology data alongside our modeled or predicted data to better inform what chemicals should undergo additional screening to ultimately reduce the uncertainty associated with their toxicity. And so a good example here would be in this study, we measured PFAS, but we also measured the slightly modified PFAS where we have the substitution of 1 hydrogen. And in this study, we measured PFAS well above what would be considered the MCL level in these groundwater samples, but our 1H PFAS was also measured at relatively high concentrations. We don't have any toxicity data that's robust for this 1H PFAS, so our default would be to rely on the data that's available for PFAS. And maybe that value is really close to the MCL level. But because we don't have strong toxicity data, we don't know whether or not this measurement is a concern at all, and whether or not we should be treating it the same way that we do as PFAS for risk levels. So at Chemical Insights, we're looking to try and utilize this data to better support our interpretations of toxicity measurements. So with that, I'll wrap up, say thanks for listening. I'm happy to answer any questions and really happy that I got to be a part of this work. It was one of my joys while I was at EPA. So thanks, everyone.
Thank you so much, Heather. That was an absolutely fascinating talk. I'm sure we have lots of folks with questions. So if people want to go ahead and drop them in the chat, feel free to do that. Or if you would like to, raise your hand, unmute, feel free to do that as well. Mark.
Yeah, I can get us going on questions while people
Awesome.
think of their own. Heather, that was a really great presentation. I learned a lot on that. One of the questions I have, and one of the concerns I have, is that with collection of so much data in NTA, I would think that it would considerably raise the possibility of false positive acquisitions. I'm thinking back to some of the earlier research on perfluorinated chemicals when it was reported that there were a lot of perfluorinated chemicals detected in food products, only to have it retracted later because there was a false positive that actually turned out to be an egg, a chemical that was present in eggs. And I would think that with NTA, that concern would go up considerably, even with the provisions that you articulated earlier about, confidence levels. Can you speak a little bit to that? Because I think it's an important consideration.
Yeah, of course. So excellent question and excellent feedback or callback to kind of historically some of the PFAS analysis. So traditional targeted analysis methods suffer from interferences. So our traditional targeted analysis methods use low resolution mass spectrometers. Instead of being able to measure out to the 4th decimal place, they typically measure out to the 1st decimal place. And so that means a lot of things, instead of having 2 clearly defined peaks, they start overlapping a little bit. And so what Mark is referencing is some early reports for, I believe, PFBA, the shorter chain carboxylic acid PFAS, where it was reported to be detected. I think the biggest study, it was chocolate cake purchased from the grocery store, and it was found to be present at very high concentrations, and people were quite concerned about it. And I want to say that work actually originated with a poster given by the FDA. And then, as it turned out, when they looked at it in more detail in follow-up studies, what they found out was that, hey, that's not there at all. It's this interferent. The same thing has been observed with PFAS and fish. So if you do a lot of fish fillet or other type of fish work, you'll know TDCA is a common interference for PFAS. And so if you're not doing your analytical work carefully, you might report really high PFAS concentrations when it's actually TDCA. The beauty of those studies was actually how they were resolved and how they were identified that it was not PFAS or PFBA was actually using high resolution mass spectrometry. So they switched to these instruments that we utilized in non-targeted analysis. And it was through non-targeted analysis that they were able to identify that, in fact, it was not PFAS, it was not PFBA, but rather it was their interferences. Now, that's a benefit of non-targeted analysis. You're able to look more closely at the mass. When they looked on them with high-resolution mass spectrometers, they were able to see the differences in those masses and say, hey, this isn't this molecule, it's this one. The downside, as you're kind of highlighting, is that now you have a whole new world of possibilities of things it could be, and does that create the possibility for false positives? Certainly when we are not good stewards of non-targeted analysis data, let me flip back. There was a quick way for me to get there. I'll just click through. When we're not good stewards of non-targeted analysis data, and we're not applying the Szymanski scale appropriately, we can get false positives. That's true of kind of any of these studies where we're relying on reference data or publicly available data that we don't have in-house, that we can confirm it with our own eyes and with our own measurements. I'll say for the work that we do with PFAS, the Szymanski scale that we utilize has been adapted specifically for PFAS analysis. And so you can see here we have this diagnostic fragments and homologous series presence. And so typically for PFAS work, we rely on a few different weights of information to say, hey, I'm pretty sure this is a PFAS. And beyond just saying this is this exact structure, we can say this is definitively fluorinated. Depending on how you want to utilize this data, knowing that it is a PFAS alone may be helpful beyond knowing the individual structure. And so I'd say PFAS is a little bit more unique in the sense that we can rely on weights of information to help us say, hey, this is definitely a fluorinated compound that is not anything that's natural. It's a man-made chemical. It has this fluorinated backbone presence. And that gives us a little bit more that we can say about it in terms of risk or hazard beyond just a non-fluorinated chemical. So it's not a perfect approach, and that's why we have to develop these confidence scales. But we do try to use weights of information to suggest when we have uncertainty versus when we don't. So good question.
All right, we have a question in the chat from Justin. And then Liz, I will get to you. How many of the 210 unique PFAS identified in the study can confidently be attributed to 3M via the electrochemical fluorination process or other attributes unique to 3M?
Sure. So a lot of our attribution of it being electrochemical fluorination and us being able to say confidently that it is tied to 3M comes from not just the chemicals that we identified. So anything that is going to be PFAS or PFCAs, PFCAs can be made through the same electrochemical fluorination process. A lot of those, you know, I would say nearly all of the chemicals that we identified can be tied to that electrochemical fluorination process. The reason we feel confident saying that 3M is the polluter in this scenario, and the reason that we believe it to be 3M's electrochemical fluorination process, is because of the context surrounding the sites. Historic waste sites that received waste from the 40s through 60s, 3M was the only one doing electrochemical fluorination at that point in time. Folks that are familiar with DuPont, they had other processes, and importantly, they're not geographically located within Minnesota. And so most, if not all, of the chemicals we identified would be tied to a historic electrochemical fluorination process. And that's not to say that you couldn't generate the molecules in other ways, but based on the distribution, the amounts that we're seeing, the ratios we're seeing them in, we feel pretty good that it's tied to that. But good question, and it's not something that's an exacting science. We have to use the observations of the chemicals we make with the historical context of the sites to come to that conclusion.
Awesome, thank you. All right, Liz.
Hi, this actually might kind of overlap or build a little bit on Justin's question that he just asked. So one of the things that makes the East Metro unique, right, is we historically knew about the 4 disposal locations going into this work and providing the data and doing this awesome analysis that you shared with us today. I guess what I'm wondering, well, as part of your presentation, you said you focused on Oakdale and Washington County landfill, and then at one point you made a reference to possibly even a 3rd source, I believe. And so kind of thinking more about that and recognizing we're also doing PFAS investigations elsewhere in the state. Can you elaborate on NTA and its use and how it could be applied as we're looking elsewhere in the state, and maybe we don't always know of one or multiple sources going into that investigation, kind of compared to East Metro, where we've known about these sources for decades now?
Sure, great question. So I'll start with the same caveat that I tell everyone at Chemical Insights as I'm getting our non-targeted analysis workflows going, which is NTA is great, but the downside of NTA is that I could spend 6 months on a project just looking at the data and trying to split hairs about bond location and things like that. And so NTA studies work best when you have a really focused study design at the start. So that way I know when I need to be splitting hairs versus when maybe a study goal isn't of interest. And so in this study, we had these really focused goals, right? We know these sites are impacted by PFAS. We want to describe more clearly what other PFAS might be present because of the historical context. And we want to be able to say, do they move through the watershed? But that's the only real limitation. In the Entails toolkit that we have online, we walk through kind of a handful of different case studies of different types of NTA studies that you might be interested in doing. They can range all the way from simple, hey, let's see what chemicals are present here, to, hey, we know, a good example is if you're familiar with tire wear products, 6-PPE quinone. That actually started because they had observable effects in wildlife, and they said we want to be able to identify what might be causing this impact. And so NTA studies can kind of range in their scope and their efficacy, but in terms of other studies that maybe Minnesota might like to do, for example, there are ongoing PFAS pollution within Minnesota. Obviously, you know that with respect to Cottage Grove. There is a lot of folks at EPA who are still working on the Cottage Grove work, and I think some people on this call likely know who I'm talking about back at EPA. But so there's a lot of use cases for non-targeted analysis. Not every single one is correct. A good example of that would be if you had a site that was only impacted by just PFAS contamination for some reason. You had a user of just PFAS back in the 60s and they never touched a different type of PFAS. Non-targeted analysis likely isn't going to tell you much more than what was there. So there's many times where we pick up a study and we use non-targeted analysis and we don't actually get anything additional or new out of that beyond, hey, these chemicals that you measured through targeted are there. So there's real boundaries that you want to set when deciding whether or not NTA is correct. But my best advice is having these really focused study goals and a clear historical context for why non-targeted analysis may make sense. So good question.
I'm not seeing anything else in the chat right now, but if history tells me anything, as soon as I say that, someone will raise their hand or put something in the chat. So we can give folks a second. Yeah, Fox.
I'll go. Thank you so much for this presentation. This is awesome. One of the things that you talked about was identifying new chemistries and going through the process of getting those their own CAS numbers and things like that, which sounds super cool, honestly. Can you talk about like what the relationship between, or like maybe elaborate a little bit on how NTA can be used to kind of end up with reference standards and things like that? Like how can we go from NTA to having a targeted analysis available?
Yeah, great question. The best example I have for this is GenX. So I'm in North Carolina. If you're up on your GenX history, GenX was first identified through non-targeted analysis studies of the Cape Fear River here in North Carolina. The Cape Fear River and watershed is impacted by historic manufacturing and ongoing manufacturing of PFAS by DuPont/Chemours, whatever you want to call it today. And that chemical was only described initially through non-targeted analysis. But if you do targeted analysis, you're like, hey, I measure Gen X all the time. That's part of EPA method 1633. And so through the identification of Gen X in our waters, that promoted that chemical to be included in future studies. To include it in future studies for targeted analysis, you obviously need an analytical standard. And so it's not uncommon for novel species of PFAS or other chemicals that are identified through non-targeted analysis to then be promoted for future targeted screening and then have analytical standards available to them. If my memory serves me, when Gen X was first identified and first began to be included in routine targeted measurements, it was only available as a standard through Chemours. I believe Chemours was required to supply standards as part of a lawsuit settlement, but my memory could be wrong. Nowadays, you can buy Gen X as a standard from other places and get it in your Wellington mix. It's readily available. And so for chemicals that are identified through NTA that have some kind of significant environmental presence, Gen X is one of them. There are pathways to say, hey, I identified this chemical, and either the company who makes the chemical from scratch here, 3M in this example, or analytical standard companies are able to then synthesize that chemical specifically for research use. So there are pathways once we've identified a chemical through NTA to make it part of our routine targeted analysis workflows. And that, of course, makes it much easier to measure that chemical in the environment if we can throw it on a targeted method and not have to spend a lot of time and energy to measure it with NTA methods. So not unprecedented. Good question.
I guess I have maybe like a higher level zoomed out question while folks think of things to ask. We've talked about, kind of the applications in source identification, things like that. Are there any other kind of up and coming NTA applications that you know of that we really haven't touched on today?
Sure. So some of the work that I'm proposing at Chemical Insights that I'm really interested in is more in the effects directed analysis space. Some folks might be familiar with that term in like old school talk spaces. But there was ongoing work at EPA that said, hey, we're exposed to mixtures of PFAS at a time, not individual PFAS. Can we take these more complex mixtures of PFAS and measure them against traditional toxicology screening assays and see if there's an effect before we try to go in and decide the individual toxicity associated with each individual PFAS? And the process there is to say, if we don't measure an effect with this mixture of PFAS, then we're not going to measure an effect likely with the individual PFAS. And so something I'm really interested in is being able to utilize our non-targeted analysis data, but also the samples that we measure with non-targeted analysis as a means to inform more rapid tox screenings and a more effects-directed analysis type pipeline. So that's my hope for NTA in the broader picture. NTA right now is still predominantly used for these one-off investigations where, hey, we have this known source, let's investigate it more deeply. But as we work through kind of the standardization and harmonization of NTA from lab to lab, my hope is that we can use it for these more workflows that allow us to better prioritize chemical contaminants for toxicology and exposure screening so that we can really get at what people are exposed to and how it's potentially hazardous to them. So yeah, that's my hopes.
Awesome. Thank you.
Yeah.
All right. Lauren asked, how many labs are there that are doing this NTA work?
So non-target analysis has a strong foothold in a lot of academic laboratories. So pick any large academic institution, they're likely to have one or two NTA researchers who utilize these instruments and these techniques. You can find it, or you historically could find it at a lot of the federal research institutions. I'll say from EPA's perspective, there's a lot of NTA folks that are still there, though as the last year has shown us, kind of the capacity to do federal research is somewhat stiffened. Then when we move into, I think someone's already said it, there are some contract labs who do non-targeted analysis. Eurofins is one of them. As part of this work with Minnesota, it was actually a collaborative project with 3 different states. So we also worked with California State Water Resources Control Board, and they have a fabulous contractor who does non-targeted analysis. I'll point out that contract level non-targeted analysis is very expensive. And so take what you traditionally or pay for a single sample through targeted analysis and apply a 10-time multiplier to that, and that may be where you start with non-targeted analysis. And the reason for that price increase is definitively because of the time that it takes to process and analyze that data. And then there's us at Chemical Insights and other nonprofit-based research organizations who do non-targeted analysis. And new to the field and where I'm really interested in collaborating and working with is folks like you guys, people who work in public health departments. So Minnesota Department of Health is a great example, who have the instrumentation and are kind of foraying into the space. So it's slowly trickling away from just research applications to more let's answer these focused research goals, and I'm very excited about that. And yeah, that price, that's a good price. Some other labs charge a lot more than that. So if you're interested in non-targeted analysis, I definitely recommend trying to identify academic researchers or people like us that work in the nonprofit space and work through agreements, because per sample it can get very expensive. And if you want to do a really robust NTA study, that you're talking about a very large contract package. So
Yeah, that's helpful context. All right. I'm not seeing anything else in the chat or any hands raised. So I think we might be able to wrap up a little bit early this morning. I say that as hesitantly as possible because usually this is the time when another question comes in the chat. But having given people many chances, I'll just say thank you so much for your time today, Heather. This was
thanks for the invite.
Fascinating talk. Really great to hear about work that partially originated in the Twin Cities area. And yeah, just really appreciate your time and expertise. So thank you so much.
Yeah, thanks everyone.
And enjoy the rest of your day, everybody.
Thank you.
Thank you.